Did you know that the human body is inhabited by trillions of bacteria? In fact, scientists estimate that there are about 10 times as many bacterial cells on the human body as there are human body cells. So, before you are too grossed out by this germy prospect, I should tell you that our microbial counterparts play a foundational role in keeping us healthy. They help us by absorbing nutrients, by making enzymes, and preventing other pathogenic bacteria from inhabiting our body and making us sick. So my research this summer specifically looked into the naturally occurring bacteria in our body and their potential at preventing urinary tract infections from happening. So, my name's Lydia Delaney, and I worked with my mentor, Dr. Sysoeva, this summer at the Department of Biological Sciences. And we explored the potential of Commensal Lactobacilli to inhibit uropathogenic bacteria. So basically what that means is that we wanted to see if the naturally occurring lactobacilli, which were directly cultured from patients' bladders, could work to prevent pathogenic bacteria such as E. coli from growing and causing infections in the urinary tract. So this summer, I studied the interactions between the probiotic bacteria lactobacilli and various strains of E. coli to determine what could cause these interactions that some scientists had observed earlier. To better understand these interactions, a genome annotation software was utilized to make predictions, and various inhibition assays were run to observe the interactions between the lactobacilli and various strains of uropathogenic bacteria. Ultimately, this research will be foundational in developing an alternative treatment to antibiotics for urinary tract infections. So, to begin, we started with a bioinformatic analysis. To determine whether the Lactobacillus gasseri and Lactobacillus delbrueckii had genes encoding for bacteriocins, the genome annotation software Bagel4 was utilized to scan the genomes of these Lactobacillus strains. After discovering two bacteriocins, helveticin and enterolysin, in Lactobacillus gasseri and delbrueckii, plans were developed to grow these strains in the lab and observe their interactions with various strains of E. coli. At this point, you might be wondering, why do bacteriocins matter? And bacteriocins are basically small peptides or proteins that bacteria can produce that have antimicrobial activity. So that's why we were interested in them. So as you can see, we had the Bagel4 doing our bacteriocin search, and then we used a Proca software to do our genome analysis. And then over on the right-hand side, you can see that helveticin was found in the search. So moving on, we went on to the experimental analysis where we were looking at several environmental growth conditions. So we looked at different types of media such as MRS agar, MRS broth, LB broth, and LB agar, and we looked at some BHI agar as well. And we tested out using parafilm to tightly seal the plate, and we also tested out using some Ziploc baggies to determine what would facilitate the growth of the lactobacillus strains and the E. coli really well for experiments. Moving on, we also did an experiment where we had various strains and plates, and we started our basic inhibition assay where we had a lawn, usually E. coli, but sometimes lactobacillus, each of the strains. And then in the top well, we would have a control, and that's where we would put the same E. coli or the same S1 or S9 as we had in the lawn, so that we would expect no interaction between what's growing in the well as with what's growing on the lawn. And in the bottom two wells, as you can see, we would put the two other bacteria that were not on the lawn to see if there would be interactions around those wells. And then moving on, we did experiments. Once we had determined that there was an interaction between the S1 and S9 and a laboratory strain of E. coli, we decided to do experiments with some uropathogenic E. coli, which do cause urinary tract infections. So we used three strains, SL45, SL46, and SL47. And then moving on, we looked to see if other lactobacilli could be having similar effects as the two strains that we had looked at, the Gasseri and Delbrueckii. And then we also looked to see if the S1 and S9 strains of lactobacillus could have inhibition effects on multi-drug resistant uropathogenic bacteria as well. And then finally, we looked to do some experiments to determine what could be the cause of these interactions that we were observing. So to begin, I want to explain what an inhibition assay is, because that's kind of the foundation of what our experiments were made of. So first of all, you have a lawn growing on your MRS media of agar. And so the lawn was made up of 10 microliters of overnight solution of E. coli usually. And then along with the lawn, you had 3 wells, which we filled with 60 microliters of lactobacillus S1, S9, and then whatever your control of the lawn was. And then we also were looking for inhibition zones around the wells, where the products of the lactobacilli could be seen preventing the growth of the pathogen. So then moving on, we saw that lactobacillus S1 and S9 definitely inhibited the laboratory strains of E. coli. So as I said earlier, we first did experiments to determine how we could optimally grow both the S1 and S9 and the E. coli together on plates. And we found that sheep blood agar was really helpful to streak out our initial freezer stocks on so that we could grow it from there. And that worked out really well for us. And we also found that MRS broth and MRS agar were both really good at growing the lactobacillus strains. And we also found that it was really important to either seal the plates with parafilm or to put the plates into an airtight Ziploc baggie. And that facilitated the growth of the lactobacillus. And then we moved on to our initial inhibition assays, where we did our basic lawn of E. coli and our 2 strains of S1 and S9 in the wells. And as I said, the S1 and the S9 both inhibited the laboratory strains of E. coli. And at this point, we determined that we just wanted to use the MRS media of agar to continue our experiments. We also found that the lactobacilli inhibit multiple uropathogens. So we found that in our first set of experiments, the S1 and the S9 both were found to inhibit all 3 of the strains of uropathogenic E. coli, which cause the urinary tract infections. And we were really excited about that, and based on those experiments, we wanted to see if possibly the S1 and S9 strains of lactobacillus could have the same interactions to inhibit multi-drug resistant strains of pathogens. And so we utilized some extended spectrum beta-lactamase producing E. coli, some Klebsiella species, and some Enterococcus faecalis species and strains. And we used multiple strains of each of these, and we found that both S1 and S9 were able to inhibit all of the strains that we tested them out on, which is really exciting considering these are multi-drug resistant, strong pathogens. And so we were excited to see something as safe as lactobacillus really attacking these multi-drug resistant pathogens. Moving on, we saw that multiple strains of lactobacillus showed potential for inhibition, which means that along with our S1 and S9, we found that other strains of lactobacillus had similar or even greater interactions with the E. coli, and that was very exciting to us. And then finally, the last experiment that we did was a realistic environment experiment, and this basically transitioned our work that we had done all summer on solid media such as the MRS agar, and we transitioned it to a liquid media interaction, and so this would make it more similar to what you would see in the bladder. So basically, we formulated several mixtures in several different concentrations of E. coli and the lactobacillus, and we had different concentrations. And over on the left-hand side, we had 7 samples. So sample 1, 2, 3, 4, 5, 6, and 7 going across. And then we made 7 dilutions of these samples going down, 1 through 7. And then once we had made these samples and then dilutions from the samples, we went ahead and plated the dilutions onto our plates right away to have like a control set of plates. So this was prior to our overnight interaction of Escherichia coli and Lactobacillus. And then we allowed the remaining solutions of samples 1 through 7 to set and interact overnight. And then the following morning, we plated and prepared several serial dilutions, as we had done initially, and we allowed those plates to grow overnight to see what would be left of what had interacted overnight in the broth. And we found that the lactobacillus had killed all of the E. coli that it was in solutions with. The only growth that survived, which are in the sample 4, were the E. coli that had no lactobacillus in solution with it. So that was really exciting to see that the lactobacilli had killed all of the E. coli that it was in solution with. So we were really excited to see that. And then moving on, we concluded this summer that the lactobacilli grow best on blood agar and MRS agar and an MRS broth, and they grow more readily in low oxygen environments because they like more anaerobic conditions. We also found that not only Lactobacillus gasseri and Lactobacillus delbrueckii, but also numerous other strains of lactobacilli inhibit the growth of E. coli. And finally, we found that lactobacilli are effective at inhibiting multiple strains of uropathogenic bacteria that can also be multi-drug resistant. So at this point, you might be wondering, well, why is this all important? Well, it's important because lactobacilli formulations could be utilized to treat multi-drug resistant urinary tract infections, which is really important because a lot of multi-drug resistant pathogens are becoming harder and harder to treat with typical antibiotics because they've developed so many resistances in their genome. But we found that our lactobacilli were able to kill off even the multi-drug resistant pathogens. So utilizing probiotic formulations with our lactobacilli provides a much more sustainable method of treatment because you're not going to see the same resistances being formed as you see with typical standard antibiotics. So finally, and most importantly, lactobacilli could be utilized to prevent the development of urinary tract infections, which is really important because ultimately we want to prevent the urinary tract infections from occurring in the first place. So we were really excited about our findings this summer and the potential for how they could be used. So thank you so much for watching.